Stem cells. These are undifferentiated cells which have yet to become specialized. They can be split into two groups, embryonic and adult stem cells. Embryonic stem cells are found in fertilized embryos. These cells are pluripotent and they have the ability to differentiate into all cell types. Places like the bone marrow, brain, and blood contain adult stem cells. These multipotent cells can specialize to form the differentiated cells in the surrounding tissue. Stem cell research is in progress, and scientists are yet to develop a way to use them to their full potential. Research is still ongoing, but in the future, stem cells may be used to treat cancer, Parkinson's disease, Alzheimer's, and spinal cord injuries.
One of the main things that is holding back the research and developments in the field of stem cells is the ethical issues surrounding them. The first issue is religion. Many people strongly believe that by altering the cells that scientists are effectively messing with God's work. Another factor that is considered in the ethic effects is some people believe that this is classed as taking a human life by the use of embryonic stem cells. Some people simply believe it is just not natural, but for non-religious reasons. They just think it doesn't occur naturally and therefore it shouldn't happen. However, these areas are all heightened by a series of miscommunications between the scientists and the general public, this allowing for information to become misconstrued and often far from the truth.
Doctors can successfully reduce the size of a cancerous tumour, although rarely manage to fully eradicate it. New research suggests that this is due to therapies failing to eradicate a small proportion of cells found to the foot of the tumour. These are known as cancer stem cells, also known as CSCs. These cells represent 1 in a million within a tumour. The CSCs are believed to fuel tumours by creating other types of cancer cell in a similar way that ordinary stem cells produce normal tissue. This means that theoretically, by targeting these CSCs, you may be able to stop the tumour growth, stopping the cancer. However, these cells are hard to target, as they are similar to healthy stem cells. This means that any therapies killing the CSC cells would also kill the healthy cells. The research is fairly recent and is not yet fully accepted by many scientists, and there is still a large amount of research that needs to be done before any effective cancer stem cell therapies can be produced.
Parkinson's disease is a progressive neurological disorder partially caused by the degeneration of dopamine receptors in the brain. The main symptoms of the condition are split into two main categories: motor and non-motor symptoms. Motor symptoms include tremors and slow movement, while non-motor symptoms include pain and depression. As you can imagine, this condition can reduce a person's quality of life significantly, as they can no longer function as a healthy member of society. However, research into stem cells has looked a promising pathway into developing a cure for this neurological condition. Undifferentiated embryonic rat stem cells can be implanted into the rat striatum, which is part of the forebrain, and these will differentiate into dopamine neurons that can help a person with Parkinson's to regain normal behaviour. The stem cells actually spontaneously differentiate into these dopamine neurons to restore cerebral function in this animal model of the disease. These pluripotent stem cells were made in vitro from the skin of adult rats. One group of rats had stem cells implanted into their brains and the cell differentiated into ganglia and neurons, functioning as a healthy part of the rat's brain tissue. The second group, however, had been injected with a toxin to mimic a disease similar to Parkinson's, and this group of rats also had stem cells implanted, and they showed remarkable improvement. However, these tests were performed on rats, and the extent that this can be applied to humans depends on the similarity between rat and human physiology. Not as much is known about the implantation of stem cells to make this process fully safe for humans, as some scientists worry that during the reprogramming stage of stem cells, potentially the human genome could actually be disrupted.
Alzheimer's is a degenerative disease that is commonly known to affect the elderly or come about as a result of serious neurological injury. It's caused by the random death of nerve cells and the interruption of the connections between these nerve cells in the cortex of the brain, which is the area that's responsible for our behaviour, personality and memories. Research into the capability of stem cells being able to cure this disease began with an in vitro experiment which displayed the variation of embryonic and adult somatic cells and their ability to conform into a neurological cell. Further research has been carried out showing that stem cells have the ability to repair model neurological disorders. As a result of this further research, it's been found that stem cells attained from the bone marrow, known as MSCs, have a greater advantage than embryonic and neuronal stem cells, as they can be derived from a varied number of adult tissues, such as the blood. These cells are also able to penetrate through blood-brain barriers, hence invasive intracerebral surgery isn't required. Precursor cells, which can be found within the spinal cord, are also cells of interest, as their mitotic nature allows them to provide a continuous source of new neurons and glial cells, which have the prospects of replacing dying cells or cells that have been suffering from an injury. It's said that stem cells may theoretically aid in halting disease. However, because of the intricate network and communications between the nerve cells in which Alzheimer's stems from, the research into the development of such a treatment is made difficult, as a varied range of nerve cells are affected due to the nature of the disease. One issue with this research is that the treatment is still in its early stages. This means there has not been enough time in order to observe the side effects caused by the treatment. On the other hand, research has shown that stem cells can be used for the management of chronic degenerative and inflammatory neurological diseases. It's also said that various stem cell populations may have the ability to induce neuroprotection, thus subduing inflammation within the brain as well as attaining the ability to repair CNS degenerative diseases such as Alzheimer's. The success of this research can open a gateway into the development of cures for conditions such as brain damage.
In the past, when one had severe spinal injuries, it was a case of never to walk again because of the damage that could have been done to the nervous tissue. In recent studies with dogs, scientists have had a breakthrough in making dogs that have been paralyzed to walk again. After severe spinal injuries, the spinal cord has breaks, and these will make it impossible for nervous impulses to travel to the lower part of the body. But by using olfactory and shitting cells taken from the dog's nose and putting them in the breaks and fluid surrounding it, the connection was re-established and the impulses could once again be passed on to the lower body. The research has not yet been used on humans, as it is not 100% effective in trying to regain all of the bodily functions after severe spinal injury. Although the stem cell treatment made the dog to walk again, it did not bring back all of the bodily functions that were lost after the injury. And if this is the case, then in humans, such functions that were lost in dogs will also be lost, and trying to regain them will be very hard. The stem cells research has not only made it possible to help those paralyzed dogs to walk again, but it is also a stepping stone and a giant leap or a starting point in trying to ensure the public that spinal injuries will not leave people paralyzed for the rest of their lives like it used to be in the past. However, expectations should not be high, as the same treatment might not have the same effect on humans as it had on dogs. It might bring some kind of improvement, but until then it is not clear whether or not it will bring change or make one to regain control of their lower bodily functions.
In 2012, Dr. B. Gurdon and Dr. Shinya Yamanaka were awarded the Nobel Prize in Physiology and Medicine for the combined studies that found the mature differentiated cells can be reprogrammed to pluripotent stem cells.
In 1962, Gurdon's discovery that transplanting a nucleus from a differential cell into an enucleated egg could give rise to a fully functioning multicellular organism was the foundation of nuclear transfer technology. But it also showed that the cell nucleus of a differential cell had the genetic ability to control development into a range of somatic cells.
Prior to the work of Gurdon and Yamanaka, differential cells were believed to go through irreversible changes which made pluripotency incredibly difficult. A metaphor known as Waddington's epigenetic landscape likened cellular differentiation to a bull rolling down from a mountaintop. As it follows different routes down the mountain, this represents the mechanisms for specialization. The bottom of the hill represents stability. The bull returning to the peak represents the difficulty perceived at the time in returning the cell to a stem cell.
Dr. Shinya Yamanaka, previously a failed orthopedic surgeon, turned his interest to stem cell research and so took up the mantle of assistant professor at the Mara Institute of Science and Technology, where he commenced his groundbreaking research. Yamanaka was aware of published research into genes and transcription factors, such as the gene YANOG. Started with a cocktail of 24 transcription factors, by trial and error he reduced his first list to about 4 transcription factors, and the result is what was now referred to as induced pluripotent stem cells. Yamanaka expects to produce stem cells for other research companies as early as 2013, but the formula has been modified. It now uses 6 factors rather than 4.